Hi everyone, this is Mrs. Belong, and today we're going to talk about cell membranes. A cell has to exchange things and materials with its surrounding, a process that is controlled by our plasma membrane or the cell membrane. Now, these plasma membranes are what are called selectively permeable. Okay, so what does that mean? Well, it means that they allow some things in and they keep other things out. So selective permeability does come into play when we talk about our cell membrane. So certain organisms are going to have a cell wall, and cell walls serve as structure. Now, they also are selectively permeable, but they give a nice structure to the cells for the organisms that have these. And the organisms that have those would be things like plants and fungus and bacteria. Okay. Now, talking about our cell membrane, and all cells have a membrane. We talk about this in terms of it being a fluid mosaic model, okay? So that's a description which makes sense because when we look at the membranes, there's many things that are bound in it, not unlike a mosaic, and it's fluid or it's able to change. The base for this are phospholipids, and phospholipids are amphipathic molecules, which means that they have both hydrophobic and hydrophilic regions. And the things that are able to be allowed into a cell or to travel through the actual phospholipid bilayer would be very small, uncharged, non-polar substances like O2 gas or CO2 gas. The fluid mosaic model states that a membrane is a fluid structure with a mosaic of various proteins that are embedded within that membrane. Now, on the surface of the cell membrane, the proteins can be right on the surface, or they can run all the way through that membrane. These proteins can be enzymes, or they can be used for transport of things like charged or polar molecules across that actual cell membrane. Glycoproteins and glycolipids are used for cell-to-cell recognition, for signal transduction pathways, and for connection to the extracellular matrix. Cholesterol is a really important molecule that is utilized for both membrane stability and fluidity during extreme temperatures. So let's look up close at phospholipids. Okay, so as we see here, phospholipids have both a hydrophobic region as well as a hydrophilic region. So looking at the molecules that make up that top part, the hydrophilic region, they definitely have polarity, whereas the long chains of the tails here are non-polar covalent bonds. And so this is why we say that the phospholipids are amphipathic. They have both that hydrophilic head as well as the hydrophobic tails. And when we're looking at a phospholipid bilayer sheet, this whole region right in the middle here is going to be non-polar. Now, there's cholesterol, as we can see, which are bound right in here, these little green molecules here, those are cholesterol. Those actually help to affect membrane fluidity at different temperatures. At warm temperatures, cholesterol helps to restrain the movement of the phospholipids. So it kind of helps to hold those phospholipids together so that they don't get moving so much that they lose the structural integrity of that membrane. At cooler temperatures, as the phospholipids want to come closer together, that cholesterol helps to maintain fluidity by not allowing them to get too close. So it maintains that fluidity by not allowing tight packing. So while we think about cholesterol and we say, okay, cholesterol's first think about it, it's not that good of a thing for us to have, but it's incredibly important actually for our cell membranes. And we have cholesterol in every single cell membrane within our body. Now, of course, consuming things that are very high in cholesterol can lead to cardiovascular issues, but cholesterol is an incredibly important molecule for the structure and function of our cell membranes. So when we look at our cell membrane up close, there's a lot of stuff going on here. Let's first talk about some of our peripheral proteins. Okay, our peripheral proteins are bound to the surface of the cell membrane. So they don't go all the way through. So here is one of our peripheral proteins right here. And we can see that it's bound to that surface, the inner surface of the inside of the cell. For this particular one, they could also be bound to the outside of the cell as well. And then we have something called integral proteins. And integral proteins, like this guy right here, okay, go all the way through the cell membrane from one side to the other. And a good example of an integral protein might be like a protein channel that is allowing something to flow through the cell membrane from one side to the other. And a cool example of something like that might be an aquaporin. So maybe this channel right here allows for water to flow through in either direction. But it could also be for something like a small charged ion or a molecule that needs to go from one side of that membrane to the other. As we look at these integral proteins, they are folded in such a way that the hydrophobic regions of the integral proteins consist of one or more stretches of nonpolar amino acids that are most often coiled into alpha helices. Now, we talked a little bit about cholesterol in that last video. Here we have them. They are yellow in this picture here. We also have on the surface here glycoproteins and glycolipids. A most common glycoprotein that you're probably familiar with are antibodies. Membrane carbohydrates are utilized most often as cell-to-cell recognition and as a way to attach to the intercellular matrix. So here we have a couple different kinds of membrane proteins. And as we can see, these membrane proteins can be used for a variety of different things. One, they can be used for transport across the cell membrane. So right like here, we have examples of transport, both active and passive. So maybe allowing for water or some other small ion that can diffuse across the cell membrane in a facilitated way. So actually traveling through a cell membrane. Whereas right next to it here, we have, this is active transport. This is requiring ATP here in order to pump a substance across the cell membrane from an area of low concentration to high concentration. So transport can be used. We can also have enzymes. Okay, our membrane proteins can be enzymes like we see right here. So maybe there's enzymatic reactions that are occurring both either inside, most frequently inside the cell membrane, that allow for reactions to occur inside that cell. Here we have signal transduction pathways. And what that is it's a protein that accepts this little triangle right here. This would be like a hormone or specifically a ligand that is floating around a signaling molecule that binds to a protein that then causes that protein to set off a pathway within the cell. So it might turn on a gene, or signal transduction pathways can allow for the amplification of a signal to make a really big and fast response inside the cell. So proteins can trigger that. You can also have cell-to-cell recognition, like we have right here, where one cell is recognizing another cell. This could be like our immune system. Maybe this is a white blood cell checking to see if our cells are actually supposed to be there and doing what they're supposed to be doing. We can also have intercellular joining, as we have here, or attachment to that extracellular matrix, which we can see in this picture as we have our extracellular matrix up above. Aside from just a cell membrane, some organisms actually have a cell wall on the outside of their membranes. A cell wall gives them rigidity or some structural integrity, as well as kind of an extra layer of selectivity when we're talking about what can actually get through to the cell. So plants have a cell wall that is made of cellulose, and cellulose is a polymer of carbohydrates, but we cannot digest cellulose. We don't have that enzyme to break it apart, which is why cellulose ends up being fiber, some insoluble fiber, which is good for our digestive system, but we're not breaking those apart and getting the sugars from that cell wall when we consume it. Bacteria also have a cell wall around their membranes, but they are made of different chemicals than cellulose. And different bacteria actually have different kinds of cell walls around them. And in fact, there's quite a bit of diversity in the cell wall itself. But an interesting fact is that a lot of different antibiotics that you take to kill the bacteria interfere with their ability to build and maintain a cell wall. And so that is how they end up dying from that treatment. Fungi also have cell walls, but their cell wall is made up of another kind of polymer of glucose called chitin. That is all that I have to talk to you about with regards to cell membranes and cell walls. I hope you have a good day.